because the MMR vaccine contains a lot of aborted fetus debris and DNA particles.
That is a creative interpretation of the facts. I'm a scientist, I will give you the facts, and then your job is to determine if Kennedy is accurately summarizing the science. The MMR vaccine protects against 3 things: measles, mumps, and rubella, 3 viruses. Here's the problem though, you can't grow viruses on their own. They only multiply inside of cells. So how do you produce these weakened versions of viruses that you need for vaccines? You would need to have a supply of cells to use to grow the viruses. Luckily, you can grow cells very easily in a process called cell culture, where you take the cells, you give them some nutrient soup, you put them in a warm box, and then they just start to multiply. This is incredibly routine in bench science. It's the foundation of testing for all modern medicines. However, before you can grow cells in the lab, you do need a source for an original cell, and this is the first of 2 things that are important for you to understand. The way that Kennedy phrases it makes it seem like vaccine makers are collecting fetuses and throwing them into a vat of vaccine ingredients, resulting in a lot of aborted fetal debris in the final product, which is just wrong. What is true is that the specific cell that is propagated in cell culture and is used to grow some of the vaccine viruses was originally from an electively aborted fetus over 60 years ago. Some cells were taken from that fetal tissue, and ever since then, they have been cultured over decades in sterile lab environments. There is 0 further fetus involvement. So here's the second fact that's even more critical. When it's time to collect the viruses after you've grown them up, the cells are broken down and carefully purified away. That's one of the central, very important steps. You don't just take the whole mixture of viruses and cells, you purify out just the viruses, the part that you want. In fact, FDA and WHO guidelines strictly limit the contents of residual unwanted cell proteins and DNA. Further, even if there was a very tiny amount of DNA, that can't actually change your own DNA, it just gets metabolized. If changing your DNA was as easy as injecting a tiny amount of DNA, we would have figured out gene therapies a very long time ago, but our body has systems in place to prevent that from happening. It's been estimated that the vaccines that have been developed and produced using this cell line have collectively saved millions of lives. Major ethical and religious organizations, including the Vatican, have affirmed that vaccines made using these types of historical cultured cell lines are morally acceptable. And by the way, using these types of cell lines is extremely common across medicine, not just for making vaccines, but for example, making therapies for arthritis and cystic fibrosis, and for many types of cancers, these therapies are manufactured using different cell lines of varying types. But to claim that vaccines have a lot of aborted fetal tissue debris displays either ignorance or willful misrepresentation of the scientific fact.